Thank you very much for having me over here, especially talking to a room full of people that inspired me to do what I do. Is very humbling and also very stressful. So I will talk about an experimental system that I set up in the lab in order to study historical contingency. And first off, the question that I am interested in is, why is life the way it is? Why the molecules that we see today, and particularly my interests are proteins, are functioning the way they are? Were there other alternatives to their function? Are these functions the only way that we could observe them? Or if there were alternatives, what were they and why didn't that take place? Why didn't those take place? All right. Of course, studying the historical progression of life is challenging. The fossil record is incomplete. The ancestral genome sequences have been overwritten by mutations, and natural evolution occurs on long time scales. And the connections between the genotype and phenotype are often intractable. And this is why molecular evolution, in particular molecular phylogenetics, is very exciting, that we can use phylogenies and use DNA and amino acid information to study evolutionary relationships between organisms. This is an example of a phylogeny tree. And by examining the distribution of character states among species in the context of their phylogenetic relationships, we can, to a degree, access to the pattern of evolutionary change through time, even in the absence of the data from the fossils. Further, by examining the topology of phylogeny, we can infer rates of speciation, extinction, and how they change through time. So I'm going to now run you through a simple version of phylogenetic tree. And we use phylogeny trees to determine the evolutionary relatedness. And this relatedness is determined by comparing numbers of characteristics between multiple organisms. The more characteristics that the two organisms share, the two taxa, these two homologs, the closer together they are phylogenetically. And here we have a time scale of 90 million years, about 89% of similarity between these two homologs. And each node represents the common ancestor of the organisms that branch out of it. So the root indicates the common ancestor of the listed data. And this way we can use phylogeny trees to identify the past events and evolutionary pressures that shapes the modern protein. But of course, the phylogeny comes with its own limitations. That is also referred as the inability to know the truth. That we can depict the patterns of evolutionary change, and however, it provides little information on the process itself. We can know how the proteins look like, but we don't know what actual processes carried them out through time. So therefore, phylogenetic was hampered by its inability to know the truth, or the true phylogenies. In order to overcome this obstacle, there have been studies that benchmark the molecular calculations that were used to create phylogenies, particularly in early 90s, performed by David Hillis and Jim Bull, that combined computer simulations with laboratory evolution using viruses that attempted to benchmark the molecular phylogeny studies. But those studies started from today and evolved an organism in the lab in a very short amount of time, and then used computer programs and simulations in particular, and tested whether they can use phylogeny trees to predict their outcomes that they observed in the lab. And I am mostly interested in not starting from today and going into the future, but rather can we go back into the past using phylogenies and then attempt to come back to the future from the past. And we have the ancestral sequence reconstruction methodology. It's a powerful way nowadays to empirically perhaps test hypotheses about evolution and the form and function of proteins from using the deep evolutionary past. So now instead of going from the past to the future, we are going back to the ancestor and perhaps access the time points, access the nodes, and learn something about the process that shaped the protein. Ancestral sequence reconstruction was first proposed as an idea by Pauling and Zachary Kendall in 1963, and in my opinion, one of the most inspirational papers. It's based on where they suggested that perhaps we can, if we have the sequences of descendant proteins, perhaps we can then infer the sequences of ancestral proteins. And Steve Banner in 1990 and 1995, that time at the University of Florida, he basically took this vision and turned it into reality. He performed the first ancestral sequence reconstruction experiment where he resurrected 12 different forms of ribonuclease proteins, and then he explained how evolutionary reconstructions can help in the understanding of physiological function within a protein family. So, how do we travel back in time? This is the Hollywood version, right? So our version is not as exciting or messy, but made it actually maybe more messy. This is the science version. And in our case, we don't need any flux capacitor. That's my token joke for ASR. So, ancestral sequence reconstruction follows an approach similar to what linguists follow when they resurrect an ancient language. The linguists make arrangements in life's alphabet to make inferences about the past. And our goal is to use the nature's word, amino acids, and DNA in order to infer the states of the ancient molecules. This is an example from Steve Banner's book, and it's focusing on this is the phylogeny for the word snow that is constructed by linguists. So here we have the English word snow and the family that used this word and the homologue taxa that use this word. We can trace back the history of the word snow using phylogeny. And not only that, we can understand the root of this word, but we can say something about the culture that used this word. So if you needed the word snow, that means you lived where it snowed. Similarly now, replacing the cultures with bacterial cultures, we can infer the whole, wait, all right, there you go. No, there. So now we can change the words with life's alphabet and infer the ancestral states of each word in each sentence, and in this case our protein sequences. So if you have a lysine in this position, the ancestor will have a lysine, and the common ancestor will have a lysine as well. And then you will do a comparative analysis throughout the modern taxa and then infer your way back into the past. And at the tip of the tree, we have the final peptide sequence. So how do we do this? This is how we start. We start with a range of modern proteins, and we start by first aligning them. These are the sequences for the modern proteins, and we create a phylogenetic tree based on these aligned sequences. That's our number one step. So you need to have availability of the modern sequence data from whatever the protein that you want to resurrect. And then number two is, well, the next step is to reconstruct the protein sequences using the calculations that you prefer. So when Steve resurrected the ribonuclease in the early 90s, he used maximum parsimony. But nowadays, the maximum likelihood methodology is mostly preferred, and it is the site that is used the most. The best estimate of each site is most likely given all the possible sites of that protein in a modern day. So it goes through an analysis of site by site. And then number three, the third step is to synthesize these oligonucleotides and assemble these genes for ancestral protein using PCR. And again, nowadays we can send our final sequence information to a synthetic biology company, and it is $1 per site, and they synthesize this gene for us. And then they subclone it again for us, this gene, into a vector where we can now transform it inside the cell, express, meaning produce a lot of this protein inside the lab, and then move on with the subsequent purification steps and then move on with the analysis. So this is the stepwise way of reconstructing ancestral genes. Of course, ancestral sequence reconstruction also comes with a box of criticisms, that there is the inability to know the truth in a very strong way this time, that neither the sequence or the behavior of a protein from an extinct organism can be known. Within the same precision, obviously, as the sequence or behavior of a descendant living today. And there's always going to be a certain degree of inaccuracy, and benchmarking the studies with experiments is currently what we have so far in order to develop and improve our methods. And the particular interest has been on the genes so far that have functional divergence. So if the family of proteins have the same function, then there would be no point to perform ancestral sequence reconstruction, because most likely the ancestor will have the same function as well. So you are shooting for a phenotypic diversity in the descendants. However, the developments did lead performances of a lot of ancestral sequence reconstruction studies, and I'm running through a bit of examples here. We have reconstructed ancient uricases. These two papers actually came out fairly recently, where we can study ASR to resurrect genes that are not even synthesized in our body right now, proteins. In this case, it's the uricase. The lack of uricase production in mammals and in humans leads to gout disease. So the goal in this study was to use an ancient enzyme to cure the gout disease in humans, instead of the current treatment, which is taking a chimera of baboon and pig and delivering to humans for treatment. So we can use molecular phylogenetics for evolutionary medicine reasons. And this is a paper by Thornton's group, these two are, that tackles the problem of historical contingency in an in vitro system where they study the hormone receptors and then try to understand the historical contingency and the biophysical causes of mutations throughout the history of this protein's evolution. And we have an example by Belinda Chang in here where she resurrects fluorescent proteins and monitors their evolution and creates a phylogeny in the lab. And this is coming from our lab, that the clues on paleo-environmental temperature and pH, the conditions of the ancient life using ancestral sequences. In particular, my interest is to study ancestral sequence reconstruction to ask the question of what is the role of contingency in evolution. How much does the past restrict our present? And how far can we go back in time in terms of resurrection, but not only in a test tube, but I want to understand how much would these ancient genes function in an in vivo system. If we take these ancient genes and put them inside a genome, could we make an organism, a modern organism, survive, make the organism dependent on an ancient gene, and then watch its activity, watch its evolution perhaps? And of course, how much does the environmental condition, how much does the experimental condition that we set up to answer these questions is in effect here? So this is a hypothetical scheme of evolutionary patterns. As I said, I am particularly interested in replacing a modern gene with an ancient gene inside the system. So the interaction between an organism and its environment will produce a series of adaptive zones, after all. And the components of the cell not only interact with the environment, but they also interact with one another as well. It's a very complex network. It's a very complex set of interactions that is making this whole cell function. So now the goal is to remove a component of this complex interaction system, and then perhaps in the heart of this system put an ancient protein that is not supposed to be there. So now assume that this is our ancestor, natural ancestor, and this is where we are now. We are going a step back into the past and now changing the adaptive zone of the whole organism and perhaps introducing it to a wider adaptive zone to explore. Did this path that I drew here, I'm not sure if you can see, there's this path, have to happen? Can we make the organism explore a wider adaptive zone by introducing more ancient genes? And most importantly, can we empirically measure this problem? My goal was then to genomically engineer an organism and then monitor its evolution in the lab, and to primarily understand the historical constraints that shape the protein function within the context of the cell. And of course, to investigate these questions, we need a system that we can manipulate, but the system needs to also have a control, and it also should have a sufficient level of variability to permit adaptation so that we can observe and compare. And so the number one question of interest was that, would such a replacement produce a viable organism? Or are we just in a fantasy land? The model protein that I set to study is elongation factor protein. Elongation factor protein functions in the heart of the cell, in the ribosome. This is our protein. This is the ribosome. It delivers pre-amino acylated tRNA molecules into the A site of the ribosome. And it helps bacteria to string together amino acids to form other proteins. It's homologous in all terran life. All life we know has this protein. And it is essential, meaning the lack thereof leads to cell death. And it is very highly, in fact, the highest expressed protein in the cell. This was a snapshot from a movie, but I'm not going to show the movie, but just to give you an idea, this is the ribosome itself. In yellow is the mRNA, in presumably green triangle is the EFTU protein, the elongation factor protein. And in red is the protein, the peptide that's being synthesized outside of the cell. And ribosomes are the protein-making machineries of the cell. They can make hundreds of proteins in a second. As we speak, as I speak, the ribosome is producing peptides. It's a very complex molecular machinery. It's a huge molecular machinery and one of the oldest machineries in the cell. And ancestral sequence reconstruction studies helped to resurrect the ancestral elongation factor proteins as well. It is an old protein that functions in the oldest component of the cell, which is the ribosome. One interesting property of elongation factor is that it is very adaptive. So if you were to obtain elongation factor from microbial samples that you gathered from a hot spring, the adaptive property of this protein also will be pretty high. It will actually directly mimic, in terms of melting temperature, as we call, that the temperature that the protein starts to lose its activity will be directly matching with the host's environment. So the bacteria in my gut has an elongation factor that lives at 37 degrees, is adaptive at 37 degrees. So this highly adaptive property is actually also making elongation factor a very good candidate to study evolution in action in the lab. And this is the study that I'm referring to that calculates all the proteins and their correlation coefficients in terms of their adaptivity into their host. And based on this adaptive property, we refer to elongation factor as the molecular thermometer of the cell. Look at the temperature that elongation factor is the happiest, you'd say something about the conditions that this host is living in. And this is the phylogenetic tree that was constructed, that is elongation factors using the bacterial family. And here is gamma proteobacteria. This is where E. coli is. Each node again represents the common ancestor of the proteins that branch out of it. So this is our youngest ancestor in terms of the ancestor of gamma proteobacteria. It's about 500 million years old, and these trees are calibrated using molecular clock calculations of Blair-Hedges group in Penn State. And so here is the ancestor of gamma proteobacteria, about 500 million year. This is about 1.3, 1.6, and then we go to 2.6 and 3.6 billion year into the past in terms of the history of this protein, the age of elongation factor. And here I'm following the linear line as we go back in time. And what this study shown was that they basically, as I was telling you, by one, producing these proteins inside a test tube and then looking at their property, the temperature that these proteins are the maximum, they have a maximum functionality. So what happens is the youngest the protein, the lower the temperature. Remember that our body, E. coli, has a 37-degree temperature adaptation. So 39, already 2 degrees of increase in 500 million years. And then you go back in time and there is an increase in temperature. So, of course, when I show this figure, I can hear the bells ringing in Simon's head. But so what this study has shown is that they mapped the the melting temperature of the protein with the predicted temperature of the Earth, the ancient Earth. So here you are, time now, time into the past. The linear lines are the temperature values that are determined by geologists, and here are the lines that are determined by the biologists that performed this study that suggests that the Earth also was the temperature of the protein's melting temperature is matching with the Earth's temperature. So you can look at the temperature of this protein and infer the environment in which this protein survived. So if you go to 2.6 billion years old, Earth was about 66 degrees back then, so on and so forth. So this is how this study used ancestral sequence reconstruction and elongation factor to determine the conditions of the ancient Earth. And this helped me because the first part in my mind, the table of life that suggested by Stephen Jay Gould was done. That it was not the rewinding in terms of a whole tape, but maybe one song of the tape was rewound using ancestral sequence reconstruction. And the next goal is to engineer the bacterial genome with an ancient elongation factor gene that will, as we hypothesized, will put a stress on the bacteria that we do this replacement at, given that it has an altered temperature stability and altered function. I'm not showing the data for the function yet, but we also know that function-wise it's different than the modern protein. But it has to be viable because I'm a postdoc and I need a system that works. So this is the chromosome, a cartoon that shows the genome of bacteria, E. coli. It contains about 4,000 genes. I used a strain that was actually generously donated by Richard Lansky. There are 2 genes in this genome that encodes for elongation factor protein, TAF A and TAF B. I'm going to refer to them as A and B respectively. What I've done is I deleted the modern A copy from the genome and then replace the modern B copy with the ancient elongation factor, the youngest elongation factor that was 500 million years old. Given that it had the closest temperature with the ancient one, with the modern, given that it's only 2 degrees different, it's 39-degree melting temperature. And this was the outline of recombination, which is a boring detail, perhaps, but it took me about 1.5 years to do this step, so I feel like I should say it. And then I named these strains Rib from the Rib Van Winkle, where waking up from its sleep, and hopefully as most of you are at this moment, and then measured the effect of this ancient gene on the bacterial growth. So bear with me. On the y-axis, I have the strains, and then on the x-axis, I have the doubling time. How is this bacteria doing in terms of its growth? This is the wild type, the bacteria that has all the genes intact, all the modern elongation factors. It's doubling every 20-22 minutes, as most healthy E. coli does in the lab. Deletion of either copy, delta refers for deletion, of either A copy or B copy results in some change in terms of increase, in terms of an increase in the doubling time of this bacteria. And then replacement of the B copy with the ancient, now the Rib strain is now growing higher than the speed that's slower than by 2-fold. So it is really now slower than the modern bacteria. So the system is viable, yet it is not healthy. Which is not too surprising, but in a way it is, it's very good news for me that the replacement of the ancient elongation factor, the modern elongation factor with the ancient protein did cause the modern organism to be maladapted, but yet it is still able to interact with the other components of the cell that are rather fine-tuned to the conditions of the modern life. As elongation factor does, it interacts with a lot of other intercellular partners in the cell. And then, yet the ancestral component did introduce a stress on the cells, which opted me to move on to the next step, which was to experimentally evolve this strain. And this is the one slide that I can never show in actual science conferences, so I'm very excited to present it here. That there were possible scenarios for this synthetic organism to evolve in different ways. One is that be very determined that the ancient protein will adapt to the conditions of modern life exactly like the modern gene will do. Or the ancient protein will adapt in a manner that is completely different than how its descendant did, which would make us think that it's maybe more contingent than we think. Or the modern network will adapt to the ancient protein, the modern cellular network, in a manner that's identical to the ancient network, and perhaps maybe resurrect an ancient network, meaning the network of proteins that this protein is interacting with. Or there will be a completely new interaction dynamic that's going to be created through the natural selection inside the lab. So this is our the little time machine in the lab that we rewind use the sequences. We rewind the tape using sequences that are ancient and inferred using phylogenetics, and then engineer organisms with these ancient genes and replay the tape through experimental evolution in the lab. I've done different ways of experimental evolution. Here I will present the data for experimental evolution in minimal media. I must say that I cropped out a lot of data, given that you guys may not be too interested in it, but I have all the backup biochemistry slides if you want to see them. So the replay with through experimental evolution in the lab, that we have the modified bacterial genome. The populations go through bottleneck every day through serial transfer into new fresh media, and then we repeat this as necessary and then measure the fitness at the end of the experiment. So how does the experimental evolution in the lab work? That it's not a selection experiment, that we provide the environment and environment selects that, and we keep the environment constant during evolution. We provide the same amount of glucose, that's the food source, and keep the temperature constant. So there is no change in temperature either. And all evolution is carried out by purely these three core processes. And at the end of each timeline, how many days you want to evolve, we measure the fitness by competing the evolved populations with the ancestral populations. And here is the petri dish that shows the colonies that carries the genetic marker, so we can differentiate the evolved bacteria with the ancestral bacteria through a color difference at either pink or red in our competition assays. And based on how well the bacteria is doing in the system, then we determine its fitness and how well it's doing with respect to its competitor. And that is through counting literally the colonies that now made it and comparing it, however days we do the competition. If it is 2 days competition, we compare the growth ratio at day 2 with day 1. I started with initially identical 8 lines and then evolved them in parallel. I will present data for 7 lines here. One of the lines is still under investigation, it's acting funny, but currently we have evolved this over 2,000 generations and measured the fitness of these lines every 500 generations by competing them with the wild type bacteria that carries the modern protein. So the results of first competition assays. Here we have the modern bacteria. We competed with modern bacteria, so it's a result of 1. They're equal. And when we delete one of the genes from the system, there is a decrease in the fitness. And here is the ancient, I'll just refer to it as the ancient unevolved line that now has the 500 million year old gene inside its genome that competed with the wild type. So there's a 30% decrease in its fitness. Next to it is increase in doubling time. And this is now how well they're doing. At generation 500, 1000, 1500, and 2000 is shown. Each box represents a particular time point for each line, each lineage. And at generation 1000, the fitness is almost stabilized, but you can also see how rapidly these lines adapted to the condition. So the fitness went back right up. The system did not like harboring an ancient protein inside its system, an essential one no less. And then every 500 generations, I have sent whole bacteria for whole genome sequencing to map all the mutations in its 4,000 genes and ask the question of where the mutations are, where are they located, and how are they related to the protein that we engineered inside the system. And the results came. This is data for the first 1,500 generations. And these are the genes or regions that have mutations with their functions and with their frequency. So 2 out of 7 populations experience this mutation. That is in a gene called NAS-A. So I looked and looked and looked, and yes, there was no mutation in the ancient protein itself. So no mutation in the protein coding region of the ancient protein itself. However, there was a mutation on this region, which is the upstream region of the ancient protein. It is not at the protein coding region, but it is about 15 DNA sequence upstream of the ancient gene. So what is that gene doing? This is the location for the mutations. I found an old paper that suggests that this site may presumably impact the expression, the production of this gene inside the system. As it turns out, this is my data, that this mutation is increasing the mRNA expression of this gene, meaning that the system is now producing more of the ancient gene. That's one solution. The solution that's valid for 6 out of 7 lines. But then there's one line that didn't do that. So what about that line? And these are the genes that accumulated mutations in this line. There are different genes, there are a variety of different functions, a variety of different natures of mutation. And at this point, I then referred to the network of elongation factor for some guidance. And this is when I realized that as a biochemist, also I've been underestimating the complexity of biology. That this protein is not only functioning in the ribosome, it is also functioning in a variety of other places, but the ribosome itself, next to ribosome itself. So here we are. This is the network of elongation factor protein. It's a hub. It's in the center of this network. It's interacting with a lot of different proteins that involve in so many different functions themselves. And I've then focused on so each line refers to an interaction of some kind, and sometimes the nodes are also interacting within each other. So this is your hub, and these are the nodes. And focusing in on 4 partners that accumulate the mutation. All right. So here we go. We have the ancient elongation factor as the hub in this system, and I have the 4 different proteins that accumulated mutations in this line. At generation 500, a mutation is accumulated on this NAS-A protein that leads a truncation on this protein. So 27 base pairs of DNA is now truncated out of this protein. The data I don't have, I have the data, but I'm not showing you that biochemically that I tested how this mutation is affecting the binding between this NAS-A and ancient protein. And this lack of sequence here is actually making the 2 protein bind tighter. At generation 1000, we have a subcodon accumulated on a mutation that is essential for the cell. It's initiation factor 2. It is the protein that initiates the whole protein production inside the cell. And evolution gave us a version of that protein that is no longer functioning. So to me, that's very weird. Yeah, I don't know why. But we have an essential protein that's no longer functioning inside the cell that we are currently investigating. And at the same time, there's a mutation in NAS-A. I have a dramatic effect there. On NAS-A, a point mutation on NAS-A. This point mutation now makes the interaction between ancient DFTU and NAS-A looser. At generation 1500, the subcodon is gone, but then we have another mutation at HAP-A, a point mutation. That's also affecting the interaction between ancient DFTU and itself, but now also creating a novel interaction between HAP-A and NAS-A. That the 2 were not interacting before, but now they are interacting. through this mutation. So the network is getting almost more complicated. And in generation 2000, we have only the one man standing puppet, and everything else goes back to its original position, even though there is the fitness has been stabilized now. So where does this level us out? We have one problem and two possible solutions so far. That we have a single protein replaced with an ancestor, and which a replacement leads the system to experience a fitness decrease. In solution number one, system is producing more of the suboptimal protein to compensate for this fitness change. In number two, the system is rewiring its network. It's not changing the expression level of the suboptimal protein, but it is modifying the proteins that are interacting with the suboptimal partner, and particularly through deletions. So it is chopping off the fat within the network. It's cutting off interactions to increase the fitness. And nodes accumulate mutations, which then introduces changes in protein-protein interaction level. Which got me thinking or ask rather, is what is the role of selection and genetic variation in the evolution of genetic systems? So I realized that there are two different camps that aims to ask this question. In one camp, we have the evolution of regulatory sequences that appear to be the major mechanism that underlines organismal evolution. In other camp, we have the evolution of protein coding regions that is the main source in genetic variation. So, of course, the question is, what is the interplay between the expression and protein evolution? Or under what circumstances, if any, whether do they intersect? Or maybe is there a convergence on the same adaptive response regardless of what mechanism that the system goes through in order to respond to environmental changes? And finally, can we gain insight into how we might deconvolute these respective mechanisms of selection? In my system, the pragmatic view is that adaptations occur both at the gene and regulatory levels. And the view is that they do happen and they converge on the same adaptive solution, which is to modify the biochemical optimization of cellular networks. That the changes in both levels appear to be connected in a single measurable aspect of the system's behavior, the KD, the binding of the overall network that is the determinant factor in terms of the response of the system. And that is the biophysical attribute that is the major determinant. And I'm afraid I'm giving one of these unsatisfactory biologist talks that was referred yesterday in Michael Russ's talk, that there's not, give me the answer, what did happen? I cannot tell you that this way or that way. I see a lot of different things. But then when it comes to understanding the role of chance and necessity in evolution, the questions were, how much does the past restrict our present? Or how the answers to our questions depend on the genetic and environmental conditions of the system that we examine? Given that I went back one step, perhaps going back to more ancient forms would allow us to explore different adaptive zones. Maybe I didn't even come to the point where the system was ready to explore different solutions in the system, that the molecular pathways were already set. But where does this level us out in terms of the understanding the role of contingency in the system? Let's see if I can get away with this, but I feel like, okay, that both things are happening in the system in a way. That we have the both views. That we have the natural selection, yes, it's robust enough to find similar adaptations and outcomes regardless of the history. So like it is true perhaps that we see the same outcome, that there's an upregulation of the suboptimal protein. And certain patterns and biological challenges always emerge from the interplay. How can anyone can say no to this? I mean, it is true. It's a fact, and it is happening in the system. But then on the other hand, we have one lineage that did not do what the others did. That there is a historical nature of evolution that makes it is subject to contingency, and the possibilities for the future, depending on what has happened in the past and what chance events occur in the future. Yeah, the system does depend on its history, and maybe we need to stretch time back a bit further to explore different solutions in the system, because it is highly contingent on the history. And I will end with saying that the older the component, perhaps the greater the likelihood of us to find a mutational point that is capable of functional innovation, that perhaps we can switch the gears in the epistatic interactions between the mutations and let the proteins, and not only the protein that we are manipulating, but the proteins that are interacting with this protein, to explore different solutions. And from my system, the small happenstances of history may lead populations along different evolutionary paths after all. And in the system that we observed, there was a potentiated cell, there was the line 8 that took a step that was less traveled by, and that made all the difference. With that, I'm gonna end. My advisor is Eric Gaucher. He's a molecular phylogeneticist, and I have undergrads that I work with and wonderful colleagues that help me, and really, I'm really grateful to the funding of NASA that supported me for the past 4 years. I was working on this project, and I'm ready to take questions.